Alright, so as it turns out, that microscopic worm you guys can see up there above Einstein has essentially the same number of genes as he does, which really doesn't make a lot of sense because, like, you guys all know that a gene is an instruction. It is, if you like, a recipe for some specific biological outcome. So how could you possibly make a human being with a recipe book that is no larger than a worm's? It's a very interesting question, and I'm currently working on a project that's going to help us better to understand one of the ways in which we have evolved to do so. So every time a gene is switched on in one of your cells, a temporary copy is made. And it's from this copy, this transcript, that information is actually read. So that's kind of like me saying you have a recipe book, but every time you want to cook something, you photocopy the page, and then you read from the copy, and then you throw that out when you're done. Which sounds like a pretty dumb way to do things. But what it means is that you now have the ability to cut up and to rearrange the instructions on that temporary transcript so that you can make multiple different meals which are subtly or perhaps radically different from the original. And you can do this according to the seasons or according to the specific tastes of your guests without ever having to abandon that one special recipe book that your parents handed down to you. And this is exactly what our cells are doing. By producing, then cutting up and rearranging these transcripts, they can produce diverse outcomes from individual genes. And although worms are able to do this too, the extent to which the process is utilised has progressively expanded in higher organisms, especially in human beings. And it appears to be one of the very foundations of our sophistication. So I'm currently utilising a novel sequencing approach which allows me to assess the full depth of this process. So I'm essentially asking, how many different versions can and do you get from an individual gene? And how does our body know when and which of these versions to use? Already I've identified hundreds of unique original transcripts arising from previously described genes, and I've only really just started to scratch the surface. So although I'm not promising to cure cancer, diabetes, or any other major disease, it's only through the exploration of these kinds of processes that we can possibly hope to understand how it is that you can build a human being like Einstein with a mind-bogglingly complex brain from what appears to be rather a simple recipe book. And I think that this is the most interesting question in modern biology. Thank you.